Okay. Alrighty. So good afternoon, everyone. Today, we're very excited to have Dr. Genhong Cheng to share his new data with us on SARS evolution immune responses, SARS-2 evolution immune responses. And I will keep the intro short, but obviously, Genhong is very accomplished in a giant in the innate immunity field. He got his PhD from Albert Einstein College of Medicine in New York, followed by a postdoc with Dr. Baltimore, David Baltimore at Rockefeller, and then later on at MIT. I didn't know Genhong, we actually both came from Rockefeller. That's really cool. He was elected as a fellow of the American Association for the Advancement of Science and the American Academy of Microbiology. His lab has made several major contributions to the innate immunity field, including identifying several major signaling molecules involved in these pathways like interferon, NF-kappa B, and also his lab was the first to really elucidate innate immune signal transduction pathway for interferon and also the transcriptional gene program for the pathway. And obviously his lab has in the past has published really exciting mechanistic work on antiviral activities of many ISGs, interferon stimulated genes that are famous for inhibiting different types of viral infections. And so today, please take it away. Genhong, we're excited to hear about all your new studies.
Thank you, Meredith, for a nice introduction. Actually, I always thought, this kind of Zoom system is interesting. I only saw Meredith on the line, but I didn't see anyone else. I thought probably that we are only two people there. Okay, so yeah, it'd be interesting to really discuss about some of the progress we made related to the SARS-CoV-2 evolution and also innate adaptive immune response against the COVID-19. So we kind of started, let me see. Okay, yeah. We started this work quite early, actually, in the almost the beginning of last year, and it's extensively through collaboration with multiple groups are in China, including the Systems Medicine Institute and also the Chinese CDC and many other the institute as well. So we'll be able to get the first-hand patient genome sequence data, and so this is the data where we accumulated on the February 20 of last year. And as you can see, within about less than 2 months, it's the first report is on the December 29th, 2019. So, but this data were 2020, February 20. It's within 2 months, we already see these viruses are much more infectious than the SARS and the MERS. As you can see, like within 2 months, we have like 75,000 confirmed cases of SARS-CoV-2, whereas comparing to this, about 10,000 patients are accumulated in about 245 days in the case of SARS, and about 630 days are accumulated by the MERS infections. So indeed suggesting the virus are much more infectious than SARS and MERS. And today I just checked about, like half an hour ago, and we have now about 157 million confirmed cases worldwide, with about 3.3 million COVID-19 associated deaths. So this is the paper we published on the early, which is February 7th on 2020, and represented about one of the earliest comprehensive sequencing analysis genome-wide. And as you can see, the SARS-CoV-2 genome contains about 29.8 kilobases. It's a positive strand RNA virus, which encodes for 29 the individual proteins, and evolutionarily this virus is between the human SARS and also the MERS, and which but more closely to some of the bat coronavirus is much more closer to that. So this original analysis received lots of attention. So this paper and 1,300 citations already within about a year period. Therefore, the journal are very happy, and they ask us to write another commentary paper which published in the February this year. And so the title is 1 Year of SARS-CoV-2 Evolution. So this work again were through the postdoc fellow Roland in here at UCLA in close collaboration with a junior faculty in the Institute of Systems Medicine in China. And then so we analyzed about 355,000 full genome copies of the SARS-CoV-2, which contains about 20,000 in mutations. As you can see here, some of the strains, especially those later clay, which like 20 B clades, were these, about within a year already had accumulation of the 30 to 40 nucleotide within the individual strains. So that's where there's lots of mutation accumulation already, which translated to about 20 to 25 amino acid changes. Some of those changes are very dramatic impact, including, for example, switch from the clay 19 to 20, there's a major change in the spike protein called B614G changes, as well as in the changes on the N protein, which is including like a R203K and G204R mutation. So those changes were significant change there. The fitness of the virus and also speed of viral spread. So therefore, this become more dominant strains later. And then in addition to that, there's additional mutations where some of them are very, very concerning, where including those happens in the core British strains, and then the South Africa strains, as well as Brazil strains, which contain multiple mutations shared, for example, by 501, the changes mutation. So these kind of changes were not only affecting the infectivity of the virus, but also potentially able to escape some of the host immunity against the viral infection. And here again, some of the list of the individual mutations, including those S protein mutation, M protein I just mentioned about. As you can see, as soon as it appeared, mutations, they become dominant all over the world in this kind of from Oceania to the Europe, Asia, and South America, Africa, North America. So they become dominant strains already, things suggesting they are more fit and also able to more infectious potentially. And some of the mutation, this data already kind of late because, it's kind of out of date because it's kind of end by January of the 2021. Some mutation like Y5 and the N501Y mutations in the spike protein were now become much more dominant. If you analyze now, most of the strains will become those kind of 501Y mutations already. So it's things change very fast. And if you're looking at the spike protein alone, this spike protein were certainly responsible for interacting with the cellular receptor for host infections, and especially through this receptor binding domain that's directly at contact with the receptor human ACE2. So if you're looking at the mutations, as you can see, the huge number of mutations within these spike proteins, and especially in those newer, really worrisome strains, I mentioned earlier, where they have multiple mutations in a single strain of virus in this spike protein, especially in the receptor binding domain. And this kind of trends likely to go on and on again, where, this is an analysis where Lula made on top of the experimental studies were studied by Jesse Bloom from Seattle. He did the in vitro culture of SARS-CoV-2 and then looking at the different time points, collect sample, do next generation sequencing. He found a bunch of mutations were accumulated within those cultural conditions. Some of them likely would either increase the infectivity, or some other one may escape from host immune protection. So we also did an analysis with the antibody, response, like looking, and we found a bunch of potential epitope within this is actually receptor binding domain of the spike protein. There's a multiple epitope, means that they're able to potentially generate antibodies binding to these particular regions. So in the patient, if they infected with the SARS-CoV-2 virus, or even like now most of us has vaccine, would likely generate antibody against those regions as well. And down here, there's a bunch of monoclonal antibody isolated as well, against the individual epitopes. And they generally can be divided into two groups. The group which are able to directly competing with the human ACE2 for binding, that's called group 1. Another group is it's away from the receptor binding, but they likely affecting change the structure of the S protein, so affecting the infectivity as well. So the bunch of monoclonal antibody already have their isolated. So we've been looking for, first we look at the epitope against this the spike protein, especially on within this receptor binding motif. Those are the region involving direct contact with the human receptor AC2. So we found 3 peptide within this receptor binding motif. And this peptide, if we use it as an antigen, we're able to see a strong antibody production against each individual peptides in the human, using human serum from the patient, SARS-CoV-2 patient, compared to the healthy patient individual, suggesting if someone infected with SARS-CoV-2, they would generate an antibody able to bind into this individual peptide. If we use that as diagnosis, we're able to pretty much able to diagnose the infected person from the normal uninfected person, who can just easily distinguish that. So suggesting there is, we are able to produce antibody against those regions, and same thing I think would true for the vaccinated people as well. So the question is whether those antibody generated in those individual regions has indeed functions to neutralize the SARS-CoV-2, neutralize the SARS-CoV-2 virus infection, or offer any protections or not. So we end up using a method called phage display method. So we're taking the PBMC directly from the COVID-19 patient, and then we cloned their the heavy chain and light chain sequence, using PCR to taking the pore and clone the heavy chain light chain pairs in, let them express on surface of the phage, so generate a phage display library. Then we use the antigens, individual antigen receptor binding motif to fish out, and this is a monoclonal antibody. Nice about this is, as you know, generally when we generate monoclonal antibody, we would using the antigen injecting to the mice, generate the hybridoma, then screen out the mouse antibody, molecular antibody. Then we need to humanize them. It takes about 3 years to generate. But this is a directly fully human antibody directly from the patient. And it only takes us about 3 weeks to get those antibody. So it's a technology really developed very, very fast. So with this one methods, we generate a bunch of molecular antibody binding to the receptor binding domain. And then the affinity is it's under 100 nanogram per ml. It's similar to commercial available one, for example, from Regeneron, from other companies, and all of them are under, if you talk of molar range, will be nanomolar range of affinity. So it's very, very high affinity antibody. And this is one of them called E4 molecular antibody we identified. And in vitro, as you can see, this is done in our own UCLA BSL-3 facilities by Sabar. So it's the neutralization activity, EC50 we call it, is about 0.2 nanomolar range, which is very, very good molecular antibody. And we also did a through collaboration with the BSL-3 facility in China side, we inject into this molecular antibody to the mice. We are able to significantly reduce the viral load in the lung and also in the trachea. And as you can see, the pathology studies showing that they also are able to rescue the inflammatory phenotype in the lung by the SARS-CoV-2 infection. So suggesting this antibody likely able to be therapeutic as well. We went further to generate the crystal, the structure of this our antibody to interact in the complex with the receptor binding domain of the SARS-CoV-2. As you can see here, so this is the light blue, green. It's more like light blue is the light chain of our antibody, and the green one is the heavy chain of our antibody. The binding, they can directly bind into the SARS-CoV-2 receptor binding domain. Actually, as you can see, this binding is almost completely overlapping with the human the ACE2 receptor binding. And if you're looking at this view, you can also see the the binding to most of the ACE2 binding areas already, and with both heavy chain and light chain binding to those regions. If you look more in details, you would see that the heavy chain binding to some, form some strong hydrogen bond with the amino acid, including, for example, the K417 residues in the IBD domain of the SARS-CoV-2, and so this heavy chain interaction, whereas the light chain interact with the region where, for example, with the N501 of the SARS-CoV-2, and those amino acids I was showing, I mentioned to you earlier, also represent to the new mutant strains of the SARS-CoV-2, where. So therefore, we're looking to see how our antibody will be the binding are affected by different mutant strains. So, as you see, mutant strain were including this B1.17. That's the one strains mimicking the strain were from the England, whereas the B1751, it's more likely from the South Africa, whereas P1 is the Brazil strains. So compared to the wild type strains, where they have high affinity, whereas then the some mutation, for example, like mutation from the British strains, and not much affected by for our antibody binding. However, our antibody indeed bind weaker to the South Africa strain and also to the Brazil strain, and mainly we believe because of mutation in the in this K 4171 in one strain, strain versus Africa and or South Africa and also the Brazil. The major difference is this it's a K 417, which is changed to the N or changed to T. So therefore, we believe that's why there's a weaker binding, but still, although it's weaker binding, but still much better. Some other commercial ones, it's, which as you see here, molecular antibody CB6 completely abolished the binding with the mutant strain from Brazil or from the South Africa. And even the general one, some of them reduce the binding as well. We also looking up this binding assay were showing that there's some reduction for the binding. And we also did a neutralization assays where, as you can see, this is a kind of in vitro neutralization assay by looking at the RBD, purified RBD in the binding to the ACE2. So those kind of interactions mimicking the viral contact during infections. Where in the wild type strain, this is affinity different antibody, which binding with neutralization activity with the wild type strains and UK, South Africa, and Brazil strains. Where indeed were shown compared to the wild type strains, where the UK strain doesn't have much factor into the antibody, neutralization activity. However, South Africa strain and Brazil strain indeed strongly reduce the neutralization activities, especially with the strain with, let's say, including the one of the regional strain, which is FDA approved one, completely lost the neutralization activity against those, Brazil strain and some other one were also greatly reduced as well. Our E4 antibody indeed reduced compared to the wild type, the neutralization activity. And we also have another molecular antibody during our isolations. One is called A2, which have a strong binding, which not much affected compared with wild type by the either South Africa mutation or Brazil mutation. So it will likely we will need to potential combination of multiple antibody in order to protect against the different strain of the mutant. So this is a receptor binding domain mutation, and then I was more worried is also is another potential site, which is very interesting. It's a unique mutations in the SARS-CoV-2 because they have 4 amino acid insertion PRRA in the middle between the S1, which involving receptor binding, versus S2, which involving in the fusion. So this 4 amino acid insertion, as you can see from the right side, it's unique to the SARS-CoV-2, not to SARS-1, not to the some of the beta SARS, not including the one most closely relevant one, which is like share over 96% identity. Still, none of those contain 4 amino acid insertion. This insertion, interesting, is also create a potential the furin or TMPR SS2 cleavage site. So cleavage happens in this between the amino acid the 685, the R, and also the S686. This position is the cleavage. So with this increase in the insertion where cleavage increase a cleavage site, and the cleavage of this one has been reported to affect increase the infectivity. So we indeed showing that as you see, this normal prep of the S protein during the in the SARS-CoV-2 infected cells, as you see the 2 bands in there. One is full-length S protein, another one cleaved S protein. And then if we're using the enzyme inhibitors, they're able to block the cleavage, and we can use the enzyme inhibitor to inhibit the SARS-CoV-2 infection as well. And not interested, we can also use the peptide spanning the cleavage site, to compete with the viral infections. As you see, can reduce the viral infection. And especially what be interesting is you're looking at here where if we, on top of the ACE2 receptor transfecting 293 T cell, if we overexpress the TMPISS2, we can strongly increase the viral infection. And then this process can be blocked by either inhibitor of enzymes or by our peptide, suggesting this site indeed important for infection. What is interesting is that we not only found it's important for infections, but also this particular sequence, insertion site is also create a strong antigenic epitope, which is able to generate a strong antibody response. As you can see, if we compare multiple different surface peptide and found this particular one, the contains this binding, insertion sequence, able to contain most of strongest antibody production in SARS-CoV-2 patient, suggesting there is it is an epitope and an antibody able to generate the binding to this region. And indeed, we can also use this peptide spanning the fusion region as a antigen to distinguish the COVID-19 patient versus healthy individuals. If we use that as a diagnostic marker, we can easily distinguish infected person versus healthy individuals. To further identify the monoclonal antibody against these regions, we again using the COVID-19 patient phage display analysis I mentioned to you earlier. So we end up identify two molecular antibody able to bind to this region, and their affinity is about in the 4 or 1 nanomolar range. So that's pretty good antibody as well. And we did a extensive mapping and found this as again, this is a cleavage site, and we found this molecular antibody exactly bind to this 4 amino acid insertion regions. We make any mutation around in this sequence, we abolish the binding. So it's indeed a very unique molecular antibody able to bind to this insertion sequence. And functionally, we did an experiment in vivoli through collaboration with Galveston, the Pei Shi's laboratory. And so he be able to infect mice with the, let's say, with the SARS-CoV-2. And then by adding the antibody, our antibody, we're able to reduce the viral load in the lung about 10-fold. So suggesting indeed play important role in vivo to protect against SARS-CoV-2 infection. But I mentioned to Maddie earlier that we have very lot of trouble to really to prove in vitrally, mechanistically to see how this antibody able to block infection, because there's many during every prep, the result, some of those, the viral stock already been cleaved, and also some other one, even during the infection, they may cleave inside of the cells. So it's very hard to get a consistent data. So we cannot, we're afraid to show you this data. But we do believe this antibody play important role against the infections, not only because they in a mouse animal in vivo data, but also if we're using this sequence to looking at the patients, we indeed see, most of patients produce strong antibody against these regions. So we do believe we are able to generate antibody, and they likely play important role for against COVID-19 disease. So far I've been mentioned to you as 2 potential region of antibodies. Already generated 1 region of antibody able to bind into the receptor binding domain, and we identified a bunch of those antibody. They likely able to compete with AC2 or for the binding to the IBD domain of the S protein, therefore block infections. So those one which I show you several example already. also showing you example that we found another epitope in the serine and the TMPRSS2 cleavage site, and this antibody are able to generate during the in the patients with SARS-CoV-2 infection, likely also able to generate in a vaccinated patient individual as well. And we do believe this antibody able to protect us from the SARS-CoV-2 infection as well. So we do have molecular antibody directly bind to this region, exactly the insertion sequences. And if any of our colleagues have a way to analyze those, the furin cleavage sites, and we are welcome to collaborate with any of you to identify the functions of this and mechanism of these actions as well. So that's something which I'm really hoping to have additional collaborations. And so far it's the adaptive immune response, especially antibody response against those regions. But as you know, my lab actually, the main focus is on the innate immune response, especially the type 1 interferon mediated gene programs as a sort of like a major, innate immune response against viral infection. In the past, almost 20 years, we've been published, numerous papers, and some of them listed on the right side on this type 1 interferon mediated signal transduction pathways, not only looking at the how type 1 interferon is induced in response to different bacterial viral infections, but also especially signal transduction pathways, but also try to understand how type 1 interferon able to go through JAK-STAT pathway to change the downstream gene expression, affecting the host innate immune response against the viral infection. So I will just give you a couple of examples, mainly two examples to see. One is the interferon inducible gene, another one is the interferon down-regular genes to see how they used to against the viral infection, especially SARS-CoV-2. So we did multiple years ago, now it's almost 10 years ago, a experiment where like unbiasedly to screening for potential interferon inducible gene has antiviral functions. As you know, interferon able to induce about, I think it's over 300 genes in any given cells, but adding all together, maybe we have about 2,000 interferon inducible genes, but not all of them has antiviral functions. And so we were looking, and also not only, even antiviral genes may not work for every virus. So we're looking for kind of unbiased screening with the individual virus to see which interferon inducible gene contain antiviral function against the individual virus. So this kind of like using a way where we clone the 300 interferon inducible gene, individually transfected into 293 T cells, then we infect the virus to see which one are able to reduce viral infection. Certainly, we are interested in those who have strong inhibitory effect against any particular viral infection, but however, some of the interferon inducible gene may even increase viral infection. So we've been through this process, we identify a bunch of interferon inducible gene which are able to inhibit the individual virus or commonly inhibit multiple different virus. So now I will look at over about 10 different viruses already. So we know some interferon inducible gene are specifically able to inhibit that particular type or particular virus, and or some of them able to generate inhibit multiple different kind of virus, more broad spectrum antiviral functions also. So I'm just going to give you example of one of them we studied more extensively. It's on top of this commonly antiviral activity. This is called CH25H, and it stands for cholesterol 25 hydroxylase. It's enzymes able to convert cholesterol into 25 hydroxy cholesterol by adding the hydroxy group at carbon number 25. So interesting, it used to be sort of like as a metabolic molecules, involving in the when cholesterol level is high enough, and then the more these molecules produce that potentially inhibit the biosynthesis of the cholesterol as well. So kind of it's a molecule involving metabolic process control the level of cholesterol synthesis. However, interestingly, this molecule is interferon-inducible gene, and it's induced during viral infection and through interferon pathway. And then during viral infection, then there's more enzymes produced, then you have more 25-HTC produced. We can detect in this 25-HTC in the serum of the patient with or mice infected with virus. And then we later showing that this molecule able to integrate into plasma membrane, able to block viral membrane and host membrane fusion. So that's where we think it's potentially play important role against the viral entry. So we did an experiment where if we taking the enzyme knockout CH25H knockout mice compared with wild type mice, we found that these knockout mice indeed have more viral infections. This virus, we're just using the MHV68 as the example, but we did multiple different viruses already. If we use a small molecule 25-HC, and which certainly it's a naturally produced molecules during viral infection, we found that able to strongly inhibit almost all the envelope of virus we tested so far. And now we have tested more than like 40 different viruses already. And those are including some of BSL-4 viruses like Nipah virus and Ebola virus, and including some HIV. This humanized mouse model, we're showing that 25-HC able to strongly inhibit the HIV viral loader and able to increase CD4 level as well. And we did a series studies, including the in vitro study and mouse model, monkey model, human brain organoids, and in the case of Zika infection, as you can see here, they are able to inhibit in vitrally viral application in vivo in the mouse model, and the monkey models done in collaboration with a group in Beijing. And then the brain organoids is in collaboration with Ben's lab at UCLA, showing that all cases 25-HTC is a strong inhibitor, potent inhibitor of Zika infection. We also, this is the first actually the antiviral agent able to protect Zika-induced microcephaly in animal models. As you can see, this is an animal model of Zika-induced microcephaly by injecting Zika into the embryos, and see there's a strong increase in the brain of the viral infections. If we add in the 25-HTC feed into the mother, are we able to inhibit the Zika infection in the brain, therefore, and reduce the cell apoptosis in the brain, and therefore the brain will become thicker, the membrane, and then end up with the normal brain instead of microcephaly. So this is the example of how 25-HTC able to protect from Zika-induced microcephaly. In case of with the SARS-CoV-2 infections, so we look, we've been studying those coronavirus at least for many, many years already. And based on these studies, we've found that the host innate immune receptor, especially actually MDA5, able to recognize the RNA component from the coronavirus, including SARS-CoV-2, and then signaling through the signal transduction pathway, goes through MAV, then activated with TRAP3, TANK, those two molecules we identified, isolated many, many years ago, then leading to activation of R3 and for type 1 interferon induction. So in that case, our hosts are able to recognize coronavirus and generate a type 1 interferon response against the viral infection. But on the other hand, as a co-evolution, the species of the coronavirus also develop multiple different strategies to disrupt this signal transduction pathway to inhibit the type 1 interferon response. So one of them is It's a molecule where in the called PLP2 in the other coronavirus and called PL-PRO in the SARS-CoV-2. And this molecule itself is a D-ubiquitin enzymes and able to get rid of ubiquitins added by the CHOP3. So CHOP3 able to add ubiquitins K63 ubiquitin on the MAV, on the TBK complex on R3 as well. And then, however, this and the PL-PRO from the virus SARS-CoV-2 able to get rid of the ubiquitins and therefore block type 1 interferon inductions. And so this is how come host-path interaction will determine how strong the innate immune response. If we're looking at the cells were infected like by SARS-CoV-2, we indeed see type 1 interferon are induced, and then interferon inducible gene, including this CH25H, are induced as well. And if we looking at the functionally, if we do the cells by knocking out the interferon receptor and also interferon inducible gene, including CH25H, and indeed, if we knock out any one of those components, we see stronger SARS-CoV-2 infection than compared to the wild type cells, suggesting indeed this pathway play an important role in host defense against viral infection. And if we're looking at the level of the 25HC expression, the small molecule, and we can see, in the serum level of mice infected with SARS-CoV-2, we see more 25HC produced, and in the patient with in the COVID-19 patient, we see more 25HC produced in the serum as well. But the problem is that the accumulation of the 25HC in the patient happens very late. It's about a couple of weeks after initial infection. So it can delay the response. I think likely because the host path interaction virus may suppress our host type 1 interferon pathway, therefore, get delayed interferon response. So if they're delayed, so may not affecting too much in the natural infection process. And however, if we're using the synthetic 25HC, we're able to see we can strongly inhibit the viral infection in vitro without any toxicity. Also in vivo in the mice, if we add in 25HC, we're able to inhibit the viral load in the mice. So suggesting, we may be able to use that as an antiviral agent. So this is an example about how interferon inducible gene are able to inhibit the virus, and how can we using that, to generate small molecules able to use as antiviral activity. So with that one, then I would like to just give you this example of interferon inducible gene. I'm also going to talk about on the other side, how type 1 interferon not only induce we call ISG interferon stimulator genes for antiviral function, but also downregulating certain metabolic genes as a way of host defense against infection as well. So as you can see here, we've been not only see lots of interferon inducible gene, but also interferon downregulated genes as well. And one of them is called fatty acid synthase or FASN. As you can see, type 1 interferon able to suppress FASN expression, and that's through interferon dependent pathway. If we're using poly-IC stimulation in the wild type cells, we can see downregulation of FASN, but not in the interferon receptor knockout cells. And so that's what during viral infection, we can also see like a downregulation of FASN, and there's through multiple different viral infection that depends on type 1 interferon receptor signaling as well. So again, suggesting this molecule is downregulated by type 1 interferon during viral infection. And the first thing is interesting is the molecule were able to convert this acetyl-CoA to palmitate. It's a critical step from the short chain to long chain fatty acid synthesis. And if we're using the genetically, if we overexpress in fasting, we're able to see viral infection increase. If we downregulate it by either siRNA or knockout vaccine, we're able to reduce viral infection. We also can, adding the downstream product, the palmitate, we're able to see viral infection increase. So suggesting this pathway is essential for viral infection. And on the other hand, by downregulating this pathway through interferon-dependent mechanisms, we're able to reduce viral infection. And the mechanistically, we found at least a couple of mechanisms. One is this. If we end up using a small molecule inhibitor of fatty acid synthesis, found that this molecule able to block viral entry. So suggesting that fasting may be involving the viral entry. We also found there is a potential this pathway involving in the autophagy formations. So fasting is involving autophagy formation, since possibly because of this kind of part of membrane required for autophagy formation as well. If we're using small molecule inhibitor, able to reduce autophagy. Those are potential mechanisms related to the viral inhibit viral infection. So indeed, we've shown that this FAST inhibitor is a broad antiviral agent. We can use FAST inhibitors and to inhibit the VSV, MHV68, HSV, and MHV68, not only in vitro, but also in vivo as well. So it's a strong, broader antiviral agent. And this is a study by the Zika infections. As you can see, if we delete the fatty molecule in the cells, we're able to reduce viral infection. We can also using small molecule inhibitor, C75, to inhibit the Zika infection in vitrally, and also in the animal models, and also in the Zika-induced microcephaly model, able to reduce the viral load in the brain, able to rescue microcephaly, also in the human brain organoids models as well. But again, this is has strong anti-Zika activity. So in response to the FAST inhibitor for these SARS-CoV-2 infections, we indeed able to show this FAST inhibitor able to strongly inhibit the SARS-CoV-2 infection as measured by viral RNA and by the PFU assays. We're using 3 different FAST inhibitor. One is C75, another is EGCG, and a third one is serolanins. So 3 different ones able to inhibit the viral load by and measured by mRNA level as of PFU level. And we also have a GMP coded genetic, it's kind of recombinant SARS-CoV-2, which is convenient to looking at the viral infected cells. And in this case, if we're using C75 or EGCG, we can strongly see GFP positive cells are much reduced, by factor analysis showing here as well. So this is already study indeed suggesting this FAST inhibitor is also can function as a broad antiviral agent. So far, we've been studying many of those interferon-inducible genes and potentially showing that there's pretty much throughout the life cycle of the virus, there's a bunch of the interferon-inducible gene able to restrict the viral infection at any step of viral life cycles. And some of them we have small molecule already, like 25-HTC, and also FAST inhibitors, and some of them we still need to develop further. So with this different combination, all those we're hoping to achieve the general antiviral strategies to inhibit different type of virus. So I want to finally give you example, like importance kind of concept about the importance of generate called broad antiviral agents. Since it used to be when we generate any of the antiviral agents, we were looking for a specific viral inhibitors were so like we have like a influenza virus specific drug, we have a HIV specific drug, and so those will require any given virus or given viral protein. You need to very well understood their functions or mechanisms. Then we generate a specific inhibitor to inhibit a given virus. And then the first is taking a lot of time to develop, or, like HIV drug, how many lab or how many years you can think about to generate cocktails. And then another one is viral keeping changes. So like influenza virus, if you take years to develop influenza drug, but then takes them half a year to get influenza resistant strains. So those kind of like the problems we're dealing with now. We know that we have constantly facing emerging unpredictable viral infection, including SARS, Ebola, Zika, SARS-CoV-2. We do not know what's the next one would be. So we cannot allow us to take in such a long time to develop viral-specific drugs. So on the other hand, we know our hosts are able to generate response against all kind of different viruses. So why not learn from a host immune system to develop broad antiviral agent? The 25-HC and also fast inhibitor are those examples for broad antiviral agent to learn from our immune system. So our study were, especially when we did on the 25-HC as a Zika immediate, the microcephaly drug, where after we published, we get lots of media attentions, and also including the Tony Fauci, and specifically the able to give us a comments for that, where he mentioned that we urgently need to develop new strategy to treat and prevent Zika virus infection and other emerging viral diseases, identify immune system components with broad antiviral activity, elucidating how they work is a promising step in that direction. So that's kind of like, as an IAD director, where he were pointed interest, importance to develop a broader antiviral agent. With that, I would like to stop my talk, and but before that, I would like to acknowledge the group who made contributions to this work, or certainly will be our UCLA group, where spend a lot of effort. Now we have pretty much everyone work on the SARS-CoV-2 already, and so really thank them for hard work. And in addition to that, we have a lot of collaboration with the Institute of Systems Medicine, from the different student or faculty members over there as well. And we did our BCL3 animal work, mostly is through the Beijing Institute of Microbiology, epidemiology work. Then we also collaborated with Chinese CDC, and also with some additional hospitals. And we also collaborated with some of work with the Galveston, the P3 facility as well. So, and then finally, I'd like to thank many collaborators at UCLA for many years for their support, for their help as well. Thank you.
Great, thank you so much, Genhong. That was a really exciting talk, and quite a multinational collaborations you have there. And we'll open up the questions. So if you like to ask a question, please unmute yourself, or you can also type it in the chat box. And I guess I can probably start with the first question. I have a lot of questions actually, but I'll start with one, Genhong. Is that, it's really interesting with the CH25H work that you guys done, and all these genes, right, that you mentioned, and also FASN, they're all involved in lipid metabolism. So I'm just curious, do you think that, in addition to their anti or pro-viral effects, right, that is, mediated by interferon pathway, could it be that the virus is also trying to regulate these genes? Because usually for RNA viruses, they need to set up their replication complex, usually in the membrane compartment. That is actually also beneficial for them, sort of separate from their antiviral role.
That's certainly, I think it's very, very important where, the many of them and indeed membrane, bounder component in the host will help the viral replication, including this, the vaccine itself, which is it's also we believe it's essential for viral replications, and so it's a may function as to providing lipids for the virus, also may potentially involving the like a autophagy formation as well. So therefore, changes in the viral level of those host metabolic enzymes or proteins may potentially affecting virus infection as well. Even CH25H itself is it's a membrane bound protein located in ER. Yeah.
Cool, thank you. I think Alex has a question.
Yeah, thanks, Genhong. That's super exciting. It's really amazing how you've been a trailblazer in this field. So in that analysis that you described in the PNAS paper, where you looked for really effectors of the innate immune response, it's actually, and then you compared how effective they are to two different viruses, RNA, DNA viruses. Well, it's actually, if I look at that list now, it looks like most of those genes actually are not effectors. They're signaling proteins, RF1 and, SOX and, all things of that sort. And you have identified some effectors, but actually there's still there's still very few known, right? And so it probably is the case that there are many of these effectors don't work very effectively on their own, but they need to function in concert. And so it's difficult to identify them unless you do sort of combinatorial screens, right? So I'm wondering, my question really is, how can we get at these combinatorial effects of these anti-pathogen genes? So, we can find more and not, because otherwise we keep going back to the signaling proteins, which are the regulators of those effectors.
Yeah, I think it's very, very interesting questions. I've been keeping thinking as well. Actually, interesting is what initially our PNS paper were, if we compare the number, we use a particular set of genes where we identify in a macrophage cell by, for example, by poly-IC stimulations. So we using that particular sets like about 300 interferon-inducible gene for our, the unbiased screening assays and our assays, whereas as you know, the Charles Weiss were the advisor were, they use the kind of similar strategy, but using totally different group of ISG, where they use these based on literature, whatever accumulated different cell types, and so end up there's not that many overlapping, which is really interesting in the inducible gene. But then, both approach a similar approach by express one by one, looking for which antiviral, which ISG antivirals. And all come up with kind of, I would say by the time now we have like 10 different virals already. Each virus, for example, we may find about 30 to 50 ISG able to suppress suppress a given virus. But again, like you said, many of them are signaling molecules where, because those are commonly especially able to inhibit the individual viral for all different viruses, which is if we're looking for common one, indeed, most of them are, many of them at least are the it's the interferon signaling molecule as well. But if we look at some individual viral one, then you will find a more specific one. So whereas, on here, if I don't know whether you can see this, so on here, there's many of those already listed able to inhibit the viral replication. Yeah. And those were the effector molecules, where what I really more studied, I more like appreciate the interferon system. As you see, since the virus live inside ourselves. They use all our own components, our own machinery, everything. If any of the pathway, if you completely inhibit or using knockout, let's say, you have to have some way to compensate that in order for cell, metabolic cell survival or metabolic process happens. So therefore, interferon strategy is not to inhibit any particular step completely. They inhibit, they slow down virus step by step. Each step only probably 2 or 3-fold in suppression, but combining every step, then they're able to achieve the antiviral approach without affect, significantly affecting the cell toxicity. So that's why I really appreciate the interferon pathways.
So it's the combination of small effects that really makes a difference. Yeah. Thanks.
I wonder if I can also quickly jump in. So I agree with also what Genhong just said, but also Alex, maybe we should talk more about this later. I'd like to hear if you have some, novel ways that maybe we can do this better. But thinking about the ISG that I've been studying, right, ZAP, initially was discovered in a cDNA screen to be antiviral for retrovirus. But then not until we did another genome-wide siRNA screen, then we found the cofactor TRIM25, which is also an ISG that is absolutely essential for ZAP antiviral activity and shown for many different types of viruses. So I guess, I really appreciate your question is that, can we, or is there other ways that does not require multiple genome-wide screens, because it's a lot of work involved to identify sort of the complex in the network, because I do think that all these ISGs, they all work with other ISGs or other host factors, and together they sort of have that maximal antiviral function. So
yeah, indeed, we're always hoping one drug or one target will solve all the problem, but that's not, at least evolutionarily, that's not the ISG is strategizing through. They were more like step by step without purposes, without affecting normal cellular activity. That's what I do.
Well, the one drug that works against all viruses is interferon beta. It's just that it has a lot of side effects.
Right, but
that's the problem.
You probably don't want to take that, right?
No.
Actually, I don't have time to talk other. We've been spent actually another 20 years where we started with the found like not only virus but bacterial infection also induced type 1 interferon. So the question is why, and so actually functionally it's the opposite. If you use knockout interferon receptor, you have even more protective the against bacterial infection. So later on we found there's an additional pathway downstream of interferon, which is not antivirus, but it's anti-inflammatory. So actually, if you use, if you use this pathway, you may potentially, like if it's interferon, you may suppress the virus in vitro, but you may cause constantly immunosuppressive condition. We're getting even more kind of. You will make ourselves immunodeficient against the virus as a chronic infection model, which we did with David Brooks, or in the chronic viral infection, we did with rubber modeling in the chronic bacterial infections, and the similar situation is found to be true in the HIV infection, where too much interferon is not good for against the HIV infection. We did some with the HBV as well. So all those kind of suggesting too much interferon is not good for antivirals.
It's the balance, right?
Yeah.
Just have to be perfect amount. Great. Is there any other questions? It's amazing we still have an audience staying behind 10 minutes past. Well, thank you for a very stimulating discussion. And thank you, Genhong, for a great talk. Have a good weekend, everyone.
Bye-bye.
Bye-bye. Thanks, Genhong. Bye-bye.